I have to say, this is the first time I'm attending a meeting such as this, and I'll be leaving the meeting having learned a huge amount about how we might improve our own research. I feel somewhat fraudulent in speaking here today, because actually our group doesn't do any production of software or visualization methods. However, what this talk will do is to present the viewpoint of a user, of a bioinformatics researcher interested in transcriptional regulation, who uses a lot of different types of visualization methods in order to communicate effectively with biologists, including ourselves. Okay? What I'm going to do is just to go through some of the most common methods that are used to depict transcriptional regulatory systems, not just networks. And many of these methods we've already seen in various presentations, but what I want to do is to go through them, demonstrating how we might use them in our own work, and also talk about some of the strengths and weaknesses that we find when using these different visualization methods. And finally, during the talk, what I'll do is to present some of the problems or issues that we face as researchers when using these different methods. Okay, so I'm aware that we have a mixed audience here. A lot of you are biologists, but some of you might not be. So just to make sure we start off on the same page, just examining what transcriptional regulation is. It's the process in which DNA, or genomic DNA most commonly, is transcribed to RNA on the way to the production of proteins. Okay? And really the regulation of this step is perhaps the most important point at which protein production is controlled, and this can be done at many different levels, including at the very sort of large-scale levels of looking at DNA structure, looking at how different types of proteins interact with DNA, and also the underlying DNA sequence itself. But the aim of looking at all these different types of information really is to understand how they all, at the end of the day, control the activity of the polymerase enzyme, which converts or transcribes the DNA. Okay? So who the hell are we? What do we do? Well, our group's research focuses on two questions. Firstly, we're interested in using genome-scale data in order to understand how transcription is regulated, so to understand different types of mechanisms at all these different levels of regulation. Okay? In addition, the second question that we're really interested in is understanding how transcriptional regulation then is used to go and control different types of interesting biological behaviors, including, for example, host-pathogen interactions and fly patterning or gene expression patterning during fly development. Of course, in order to do our work, we're a computational lab. We integrate many different types of data, ranging from the basic DNA sequence to understand what these proteins are binding to, transcript levels, which measure the amount of gene or protein being expressed, and these are produced by technologies such as microarrays and others such as sequencing. We want to understand where protein DNA interactions occur, and here we use crystallographic information as well as genome-scale data sets such as ChIP-seq data. And then finally, more recently, we've been getting more and more into understanding how the nuclear organization of the genomic DNA affects transcription. And here we use data such as fish images and more recently, Hi-C and ChIP data. Now, of course, these different types of data provide very complementary information describing a particular regulatory system. And just to emphasize that although our perspective is very much of a genomic perspective, we also do look at molecular information in order to see whether these detailed observations are consistent with some of the more general observations that we make. Okay? Now, of course, as bioinformaticians, the day-to-day work we do involves looking at lots of files that look like this. Really doesn't matter whether it's DNA sequence or structural data or whatever. We look at a lot of text-based information, and the aim of our work is really to get to a nice visualization or schematic of how the system works. Okay? And there are many different steps involved. Now, I've grouped most of the visualization methods that we use in our work into 4 main categories. Now, these different methods convey different types of information about a particular regulatory system, and each style has many strengths and weaknesses. So there's no real one killer method of displaying regulatory data. And one thing that we always try and emphasize is that the method we use to present any information has to be grounded in the biology. However abstract the presentation can be, we must be able to bring it back to the biological question that we're trying to answer. So I'll go through these in turn. We're familiar with many of these presentations, but just to give you an idea of how we use them, both in order to inform ourselves about new discoveries in the data, and then also to convey these results to readers and our collaborators. So genome browser view, probably the most common view of looking at genome scale data these days. You can go to conferences where you see sort of one genome browser view after another. Basically, what you have in the horizontal axis is a segment of genomic DNA. In this case, we have the fly chromosome X. And then we have different features lined up in different rows down the y-axis. And in this case, we're looking at binding by 3 different regulatory proteins in the fly genome, and we're also displaying the binding sites with respect to the genomic annotation. Now, there's some obvious strengths to looking at genome browser views. One obvious strength is that you get a very intuitive view of the relationship of these functional genomic data sets, such as binding in relation to genomic annotations. So, for example, in this nice example from a paper taken from a colleague of mine, what we can see is that the binding sites of this transcription factor STAT1 aligns very nicely with the start sites of genes. Okay? And because of the location in which they bind, one can often infer a regulatory relationship. In this case, this gene, this transcription factor, is probably acting as an activator of many of these genes. Another strength, similar to this, is the fact that we can start aligning lots of binding patterns by many different proteins and start to investigate the relationships between these different proteins. So, for example, here we have a plot showing the binding pattern of multiple regulatory features. In the top 2 rows, we've got the pattern of covalent modifications of histone proteins along the genome. And here, these binding patterns are spread out in sort of large extended regions. Okay? So you've got continuous regions with one type of binding and other continuous regions by another type of binding. We've also got transcription factors, in this case a CTCF, and you can see here you get the traditional sort of nice narrow peak of binding in contrast. And then we can also find in this plot polymerase binding, which shows the amount of transcription that might be going on in a particular gene. So, and what we emphasize here is that you don't necessarily have to just show binding, but also exposure to nuclease, cleavage, etc. This type of view gives us the ability to display, sort of compare the binding positions by these different proteins. For example, the 2 histone modifications are very much anti-correlated with each other. What's the biological inference? Well, they're probably mutually exclusive, quite an easy one to say. whereas the polymerase and CTCF binding are correlated. And because polymerase indicates the amount of transcription, here we're probably looking at transcriptional activation, okay? Whereas when you look at the relationship between these histone modifications and polymerase activity, you might start inferring the opposite, where this type of modification is largely repressive of transcriptional activity. This type of modification is activating. And then this other one is probably conditionally activating, because you also see another binding site within the repressive area, but that's not leading to any gene activation. And actually, quite a lot of the work before we start getting into all kinds of calculation does involve just looking at the binding data to start to see whether there are any obvious patterns. Another really nice advantage of looking at genome browser views is that it scales very nicely. So here we have the binding of a fly chromosome by a nucleoporin protein, quite a small segment, 100 kbs, and this is scaled out to look at the binding pattern of this same protein across the entire fly genome. And the segment that we're looking at is just a very small area. And by looking at this binding pattern at these different scales, one can get some very different types of information regarding the binding and regulation by this nucleoporin protein. Of course, we find huge weaknesses also. The first weakness is that, as we've noted earlier, it's very much linear. So here we have fish images of the actual fly genome in the top row and in the bottom row inside the nucleus, shown in green. The X chromosome is on the right, and you can see that actually this X chromosome is nowhere near a straight line, and also actually spatially within the nucleus, and this is very important for its regulation. Spatially, it's actually very close to the wall of the nucleus rather than being in the center. Okay? So this is lost in a genome browser. Another weakness that we find, especially when we get more and more data sets available, is that we can only display a limited number of features. So in this paper here, this paper investigated nearly 40 different modifications, and this is about the limit of what you can display. Another problem that we find is that these browsers show only one-to-many relationships. What do I mean by that? Well, we can look at many different types of proteins binding, but this is concentrated on one region. And here we show two particular genes, but these occur on very tiny sections of the entire genome. So we're missing out on looking at the entire genome. What a lot of people do is then to go back and average the profiles by aligning, say, the transcription start site and plotting the average binding of these things surrounding this area. But actually, and this is very common in papers by labs that produce a lot of these data. But what you'll see in a minute is that this type of averaging also hides a lot of individual variation. So what we're seeing here is sort of advantages and disadvantages. Heat maps, we're very familiar with them. Here, as I said, we're aligning particular genomic regions and then displaying the amount of binding or whatever based relative to that position, and then the amount of binding or whatever we're representing by the color. Here, the axes are swapped from the browser, so we're displaying the aligned genes on the y-axis and different types of DNA binding proteins on the x-axis. Now, the advantage of the heat map, and this is an average signal taken from one of these papers of polymerase binding, and in blue you have the transcription start site. The average is that the advantage of this cluster or this heat map is that it spreads these signals out. And what the original authors did not show in their paper is that the averaged figure or the averaged line actually masks a huge variety in different types of binding signal by this polymerase protein. It's great for us because it gives us more analysis that we can do on public data, but it's a shame that the authors missed this in the original publication. Okay? And we can take another profile of a polymerase in a different sort of modification state. Again, you can see the average profile looks nothing like or representative of all the different binding patterns of this protein across the genome. Okay? Now, of course, you can go and start clustering these heat maps or the signals in these heat maps. And here we got some nice clusters, about 9 of them, representing sort of quite big groups of binding patterns. And then the other advantage, compared with genome browsers, is that one can start presenting quite good regulatory relationships compared with these clusters. So here we've added expression data from totally a different data set, going from low expression to high expression as you go towards the right. And we can see that these different clusters are showing different levels or different binding patterns represented in these clusters correspond to different levels of expression. Okay, so high expression, no expression. But the advantage here is that we're maintaining also the gene-by-gene mapping. Some of the weaknesses here are very similar to the weaknesses found in the genome browser view, size limit, linear. Again, the relationship is few to many. This time it's the opposite way around because you're limited in the number of DNA binding proteins that you can display on any given list of genes. Networks. So networks we've heard a lot about in the earlier talk. And here what we're doing is to interpret the information we get from transcriptional regulatory information. Okay? And we're inferring a regulatory relationship. So here we said, oh, there's probably a causal activation going on. And then what we can do is to sort of reduce this map into 2 nodes, the transcription factor and the target gene, with the arrow representing sort of an activation. Okay? And if you take many of these, you can start getting very beautiful images of entire regulatory systems. In this case, we're displaying the regulatory network for the entire yeast genome. Of course, compared with the genome browser and heat map views, one of the main advantages here is that it's extremely compact. Here, we're representing about 6,000 regulatory interactions between transcription factors and targets. And we couldn't do that possibly in a genome browser. And also, we start to overcome the limitations of just seeing one gene at a time or just a few regulators at a time because this network accommodates many-to-many mappings. Okay, so each transcription factor, we can show regulating many target genes regulated by many transcription factors. Another advantage, which was beautifully illustrated by Trey's talk, was that networks allow us to access sort of a different type of way of analyzing these regulatory systems. Okay, so one very nice example that was published quite a while ago by Urialon was to reduce this huge network into what they called motifs, which are sort of these connectivities that are found repeatedly throughout the network. So, for example, one of the motifs that they found was this one called the single input motif. And you can find examples of this where you have a single transcription factor shown here in green regulating many target genes. Single input because there's only one regulator. And there's a specific kinetic response associated with such a connectivity. So here we show the expression level of the transcription factors and target genes with time. The transcription factor is upregulating, its expression level is going up. Depending on the threshold at which the target gene is activated, you can have a time delay in the series of these target genes being activated. So the blue one has a low threshold activated early on. The red one has a higher threshold, which means it gets activated later. Feed-forward loop, here we have SBF, a transcription factor regulating PLM2 and intermediate, regulating a final target in blue. But what we also see is that the primary transcription factor also directly regulates the final target. And here we have many different types of responsives, one of which is that if the transcription factor has a transient upregulation and then a prolonged expression, the intermediate transcription factor will effectively filter out this transient input and only respond to these prolonged ones. Okay? So by breaking down this regulatory network into these motifs and looking at the component structures, we can start to understand how this network functions eventually. And by taking this whole network and representing individual motifs as these sort of icons, what Urialon and colleagues did was to decompose the entire E. coli network into these series of motifs with transcription factors feeding into a layer of multiple inputs which feed out into individual target genes. But then also in the output you have these single input motifs and feed-forward loops that connect together. And this gives a very nice representation of what could be a very complicated system into something that's quite intuitive and understandable. The final sort of strength of networks is that it interconnects with other systems very nicely. So for example, in our work, we wanted to look at how the metabolic network is regulated by the transcriptional network. And by using a network representation, we were able to bring together these two different networks, showing how different parts of the metabolic system is regulated at different times. Of course, there are weaknesses here. One is what we call the giant hairball syndrome. Our network looks very similar to a hairball, and these networks can be very far removed from the biology. And this is a very common criticism that we get when we collaborate with experimentalists. Another thing is it's not clear how you should represent all these different types of regulatory relationships into a single network. These different binding proteins bind in very different ways. And they also represent quite different types of regulatory relationships, sometimes totally repressive, totally active, or half of each. And just to finish off, of course, the spatial map is incredibly important. So the nucleus, here we showed two representations of the nucleus, one taken by fish, and each chromosome in this human nucleus is colored differently, and then a high C type representation of the yeast genome. And so these chromosomes reside in these very specific territories within the nucleus, and it's becoming clearer and clearer that the nuclear position of particular genes impacts greatly on its transcriptional state. Now, of course, the great strength is this is what the nucleus actually looks like. This is how the genome is arranged. It's beautiful. And you can do these network maps, which was originally published, showing which parts of the chromosomes come together closely together in the nucleus. And we've started to map gene expression data on these representations to see if we can identify clusters of genes that are far apart on the chromosome but close together spatially, if they're co-expressed or not. Of course, the problem here is that we're still very much at the early stages of understanding the biology. We don't really understand the relationship yet, okay? And then there's technical limitations in that what we see here is an extremely averaged view of the spatial organization of the nucleus. And some papers have reported where two loci might be close together in this averaged image. Actually, they only occur in about 6% of individual nuclei. So what I've done is to present sort of the 4 major visualization styles used, or that we use, to investigate transcriptional regulation. And this is quite common across many of our colleagues. One aspect that we find constantly is that we're switching between these different representation methods. And it's not because a lot of the software tools that we use are focused on one representation. It's not easy to go from one to the other and quickly switch between them in order to investigate the same sort of region. Okay? And in any given publication, we might be mixing 2 or 3 of these visualization methods in order to get all the different points that we want to across. The other sort of question is how could we visualize or use these visualization methods to visualize new types of information? So what I didn't really touch on in here was quantitative data, the binding strength of a transcription factor, of course, intuitively shown in these genome browsers by the height of these peaks. How do you represent that effectively in a very complicated network? The dynamics. So what we often measure are very average things, but there are changes from cell to cell. There are many changes during a time course and over the course of evolution. How can we use these different methods to represent these changes? And then, of course, how do we include spatial information, not just the nuclear organization of the genome, but say the expression pattern of a gene across an entire body, for example. And also, although we're getting into more and more about animations and using sort of interactive things, at the end of the day, most of our work is presented in journal publications and people just print them out. So how do we represent these complex information in a static way? So just to summarize, I think, and what I take away from this meeting is that we can still learn a lot about effective ways of visualizing. If you take a typical genomics paper, you see a very clear pattern. What you get is a genome view of the data to give you the sort of the surprise or the wow factor of the scale. Then you'll get an averaged view and then a single gene view. You can sort of pretty much do this automatically. But we can learn a lot about how we might present the data more effectively to convey the information that we want. Very nice visualizations can also help bring things forward further into the discovery stage of the analysis, which we don't really touch on very often. Normally, we just think of visualization, or in our work, as something you do when you want to write up a manuscript. But actually, if you get a nice visualization method, it can be very effective as a tool for discovering regulatory relationships, for example. And also, if the figure is pretty, it can make a paper memorable. And this is something that we look for. In the worst case, though, they can just confuse everyone. And this is what we always try and avoid. OK, so just to finish off, oh, yes, and also the final thing is that to be effective, any kind of visualization method, however abstract it may be, has to connect back to the biological system you're trying to look at. And without this, I think the use of the method becomes very limited. So just to acknowledge, I presented some data from papers published by group members, particularly Juanma Vaqueritas, who's a postdoc in the group, and Garth Ilsley later on as well. OK, so thank you.